Hi, my name is Callie Chappelle, and thank you for tuning into this video about mitosis and meiosis. I'm a biology student at the University of Michigan, and this is a series of videos for Bio 305, which is Introduction to Genetics. Due to the breadth of the course, I wanted to make a series of videos that highlights the most important aspects of the class that you'll need to know for an exam. I like to call this the quick and dirty. So if you're the kind of student who just wants another review, or if you're the kind of student who maybe wasn't in class, hopefully these videos will help you out. So here's the quick and dirty for this video. The first is mitosis that we're going to talk about, the second is meiosis, and the third is nondisjunction. So let's get started. Let's talk about mitosis. So we know, perhaps from 9th grade bio or perhaps later, that mitosis is where you start with something that is, let's say, 2n, and it results in something of the same ploidy level. So it also ends with 2n. So let's say we were talking about something that was just n, it would start with n and it would go to n as well. So I'm going to assume that you're already familiar with the steps of mitosis and meiosis, but we're going to really focus on what happens to alleles as they segregate, because this is important in the context of connecting what's actually happening in a cell to doing statistical analysis about the segregation of alleles in a quantitative setting. So we're going to start out with, we're going to do this in the context of 2n. So let's look at our 2n cell. So we have a 2n cell, 2n, and we have two chromosomes. Each chromosome has a different allele. So this chromosome has big A, and this chromosome has little a. All right, before mitosis can even occur, we have S phase. So what happens in S phase? Think back to the cell cycle. Replication. So we're going to have replication of these two chromosomes into two sister chromatids, also with two chromosomes. If you are a little bit confused what I'm talking about, then go back and watch the 5-minute Back to Basics video that I just did, where we talk about this in slightly more detail. So this chromosome replicates, this chromatid, aka chromosome, replicates into two sister chromatids, which are also considered, this is one chromosome. This one replicates into two sister chromatids. Here are those two sister chromatids, one chromosome. So the phases of mitosis, if you don't remember, are, oops, my pen's not tracking very well today, prophase, metaphase, anaphase, telophase. For time purposes, we're just going to go straight to metaphase. So when metaphase happens, the chromosomes line up on the metaphase plate. So here's the metaphase plate. I'm just going to label this metaphase. How do you think that these chromosomes will line up? Well, because in the end, we want cells that look identical, daughter cells that look identical to the starting or mother cell, they have to line up like this. So what does that mean? Well, our final product needs to also be 2n, and also our final product needs to result in daughter cells that have the big A allele and also the little a allele. So these are connected. I didn't draw the kinetochore. I didn't draw the centromere very well here. So here is big A, little a, little a. Now, why is this the orientation at the metaphase plate, and why is it not this? Why is it not this? Okay, so this seems equally probable, but this is not what happens. Well, if these segregated, you would result in daughter cells that were either big A, or little a. Now these, although they're the correct ploidy level, they're 2n, they do not match the genetic makeup of the alleles that were in the mother cell. So this could not be the correct orientation for mitosis only. So this is the only correct orientation. So make sure you remember that, because we're going to have some more complicated questions coming up that make sure that you know and understand that this is the orientation on the metaphase plate. So metaphase happens, we're going to have segregation of sister chromatids, and this will result in daughter cells that look like what we just talked about. Two chromosomes and with 2n ploidy level and the same genetic makeup as the mother cell over here. Okay, so this one is going to look exactly the same. Big A, little a, and also will be 2n. So make sure you understand how these alleles are segregating. So let's talk about a slightly more complicated case of meiosis. So meiosis is where you start with something, say 2n, and then you result in something that is half of that, n. So let's say we're talking about a tetraploid organism. So that would be 4n going to 2n. Cool. Okay. So what happened to these cells? So we're going to do this case where we start with diploid and go to haploid, because that is what you'll most likely see, also what happens in us. So we're going to start out with the exact same cell that we had in the meiosis slide, like that. Okay, so this starts out exactly the same, S phase and replication. So we're going to have 2 chromosomes, each with a identical sister chromatid, just like that. So the stages of meiosis, just a reminder, are prophase, metaphase, anaphase, telophase. Prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. So it's kind of like mitosis happens twice, but not exactly. That's not a perfect analogy, but one that provides a decent heuristic for understanding how this works. So again, we're going to skip straight to metaphase 1. And let's figure out how these guys line up on the metaphase plate. So this is a big distinction between meiosis 1 and mitosis. They do not line up, remember, on meiosis 1 they lined up like this. They actually line up like this. Now why is that? How could that be? Well, keep in mind the purpose of meiosis is to go from 2n to n. So if we're going from 2n to n, the daughter cells are going to look like half of that. So this is going to be n, and either it'll have big A or it'll have little a. So the problem that we ran into with this orientation for mitosis was because it would not result in cells that had the correct makeup. It would not result in cells that looked like the mother cell. However, in this case, meiosis, it doesn't matter because the daughter cells are not going to look like the mother cell anyway, so it doesn't really matter if they have, you can't have the same or different composition. Okay, so this is the way that they line up in meiosis. Now, let's see what happens. What do they produce? So here, we're going to have 2 daughter cells that are also 2n. This one, so let's do this side first, is going to have the big A, and this is going to be little a. So now, each one, now this is a little tricky. Each one of these cells has 1 chromosome. So in the previous slide, in meiosis, each cell had 1 chromosome as well, but this one has 2 sister chromatids, whereas in the in mitosis, it had 2 chromosomes, 2 chromatids. Here we have 1 chromosome and 1 chromatid. If you're getting confused with this terminology, again, go back and watch that the Back to Basics video where we talk about this in slightly more detail. So now we have these. We're ready to go to meiosis 2. So let's look at what happens at the metaphase plate of mitosis 2. So let's do this one with the little a alleles first. So here's the metaphase plate. Here is metaphase 2. How is this going to line up? Well, in this case, there's really only one way. It only can line up like this, like we saw in mitosis, and these sister chromatids will segregate like this. So likewise, if we're looking at the top one coming down here, I'm just going to make this go a little bit more. Here again is our, wow, that was a really terribly drawn metaphase plate. Forgive me, please. These are going to segregate like this. So let's look at what the final daughter cells are going to look like at the end of meiosis. Well, here, this is going to segregate into this cell, so it's just going to be little a, and that is what plate level? N. Good. Then we'll also have another one, because this will go to a separate cell, so that's also going to be N. This produce a daughter cell with just 1 chromosome with big A, that's N, and it will produce another daughter cell that looks exactly like that with just 1 chromosome and a big A. This is also, and I'm sorry, my writing has gotten just worse and worse as this has gone because I'm still learning how to use this drawing board. Please forgive me. But what is the end result? Well, we have 4 daughter cells. Each daughter cell is N. Each daughter cell has just 1 allele. Okay, so let's do a sample question to make sure we understand how all of these things work together. So this is how I would recommend working through the sample question. First, I want to see if you can do this with just the information that I've already given you in this video and with lecture, because let's say you do get the right answer, then you don't have to watch this. But if you don't and you want me to go through it, then just keep watching this video. So I'm going to say pause the video, try it yourself first, and then when we go through it together, you can see where in your thought process perhaps you went wrong, or if you got it right, that you're a really cool person. Okay. Let's consider a single germ cell with the genotype AA, BB undergoing meiosis. Which of the following are possible genotypes for the 4 gametes resulting, I don't know where this ST, or I don't know what this means, resulting from meiosis of the single germ cell, assuming that there's not any disjunction. So the first thing that we want to figure out is, what is it asking? What is it asking? Sometimes these questions can be actually a little bit complicated because you're just like unsure like where even to start. So looking at the question and underlining the most important parts is a great way to figure out what the question is asking. So consider a single germ cell with the genotype this undergoing meiosis. Well, first you have to figure out what the hell this even means, right? So we know that this dash means they are on separate chromosomes, and this means they are separate genes. So that means that if I wanted to draw this cell, what would it look like? Well, here's our cell. We know that big A and little a are on separate chromosomes, so big A, little a. And then we know, I'm actually going to do B in a different color so we can watch this. I'm going to do this in blue. And we know that B, big B, little b, are also on separate chromosomes. So if we wanted to draw this cell, actually looks like this. And we are undergoing meiosis. Well, that's good, because we're not talking about mitosis, we're talking about meiosis. All right. And then, which of the following are possible genotypes of the 4 gametes resulting from meiosis of the single germ cell? Okay, so we need to figure that out, assuming that there is no non-disjunction. So the first question is we need to figure out what we know. So what is it asking? What are the genotypes of 4 gametes? And then what do we know? Well, we kind of already just did that. We know that the cell looks like this, and we know that we're talking about meiosis. So the next question that we want to ask ourselves is, can we eliminate any, wow, it didn't cross, any answers? Well, right away, we know that we can eliminate A, because we're talking about meiosis. So we're going from something to half of that. So we're actually looking here at what kind of cell. We're actually looking at a 4N cell here. So that's kind of weird. This is not what we're used to seeing. So if we start with 4N, half of that is going to be 2N. Now let's go through and label the ploidy levels of each of these things. So this is, if we just look at this in a vacuum, that's just N. So that's like we're going from 4N to N. And so that obviously can't be correct. Now these, that is what, 2N. So that means that we're going from 4N to 2N. Okay, well that means that all of these could be correct. So looking at ploidy level just eliminated A. So let's look at C next. So in C, what can we notice? So I'm looking at this on a meta level. Here in B, D, E, actually in B and E, we have 2 things that are the same. All right, so these 2 are the same, these 2 are the same. In B, these 2 are the same, these 2 are the same. In C, they're all different. Well, how could they all be different? How could that all be different if we're all dividing in half? And we already know how meiosis happens in metaphase of meiosis 1 and anaphase of meiosis 1. The 2 sister chromatids segregate together. That means that you can never have daughter cells that are all different. Okay, so that means that at least C is out of the question, and also this also eliminates D. And then we can further eliminate answers because D and E couldn't be right because these have 2 A's. How could you have 2 A alleles and 2 B alleles? How could you get 1A and 1B from mom and dad if you actually got 2 A's from mom, right? That's not actually going to work. That means that our answers have to be A and B together for each gamete. So that means that based on what we know about how segregation of alleles actually happens. So that means that the answer has to be B, and that we eliminated answers just by knowing basic things about meiosis and mitosis, not even by actually figuring out this question. So now that we know what the answer is, B, we can actually go through and step 3, prove it to yourself. So we're going to prove it to ourselves by actually drawing out what happens. So let's go back. I'm going to put this back into green. And we're going to go through what this division actually looks like. So maybe you're dubious, and I always double guess myself. And I'm like, okay, Callie, I thought I used my smarty pants brain to figure out what the hell's going on, but then I'm like, but I probably am wrong and probably missed something along the way. So I always think it's good to double check myself anyway. So here's our starting cell, and we're going through S phase as we always do to get double of that. So remember, we're going from 4N to, golly gee, 8N. Okay, so we have replication, big A, little a, little a. Oh, I'm sorry, that was supposed to be B. Just kidding. Let's not do that. I'm not gonna do this to you with the colors because that would be really mean. I'm gonna stick with the same colors. Give me a sec. We're going to A's again. So here's the actual replication colors. A, B. All right, and then B, we're going back to blue. Oops, oh no, I messed up again. Whatever. Big B, little b. I just realized that I wrote B instead of A. These colors are just like really challenging. I honestly don't know how Sal Khan does it. He's clearly a god. All right, so now we're gonna skip straight to metaphase. How do you think that these bad boys are gonna line up on the metaphase plate? Well, we already know from our knowledge of meiosis that they are going to line up either like this, big A, little a, or like this. So this is metaphase 1. I'm going to draw the A's in the same location so you can see how they're toggling with the B allele, with the B gene. Okay, so here's the B gene now. So up here, I'm going to put big B on this side and little b on this side. But because of the law of independent assortment, guess what? It could be that the little b lines up on this side and the big B lines up on this side. And these two things will produce different products, okay? So when these segregate, the daughter cells from here will look like this. Little a, little b, little b. And this one's going to look like big A, big B, big B. And these will produce cells, and I'm going to keep in mind this is OR, so both of these things don't happen, one or the other of them happen, because as we'll talk about in statistics, OR in this case is mutually exclusive. So here we can either have big A, little b, or actually in this case little a, big B, big B. Okay, do you see how that happened? Because this is this, this is this, up here, this is this, this is this. And these two things, because they're OR, don't happen together. It's one or the other. So I'm actually going to do a huge or here. So either this happens or this happens based on the 50-50 chance of independent assortment. Okay, so let's talk about how this segregates in metaphase 2. So in this case, metaphase 2, so here's our cell, here's the metaphase plate. It's actually going to line up like this, remember? So metaphase 2, little a, little b, and then it'll segregate like this to result in 2 products. I know, this is a huge diagram. I don't know what I've gotten myself into. It's crazy. Little a, little b, right? And this is 2n, because started out with 4n, it's meiosis, go into half of that, 2n. This will segregate like this. Big A, big B, little b. Right, and this will result in 2n. I'm not going to draw out what the actual cell looks like, but I'm going to write what the genotype is. 2n, A, B. And now that we're a pro at this, can you guess what the gene products are going to be for these? If you guess 2n, big A, little b, you'd be right. And then 2n, little a, big B, little a, big B, okay? So let's look at these in 2 different colors. I'm going to choose like a really obnoxious color because I really want to make sure that you can see the difference. So the products of this can either be this, so that's little a, little b, big B, or this. Let's use a different color so we can really see the difference. I'm going to do this in orange. Hopefully you can see a difference here in those colors. Yeah, that's pretty good. This, okay? So let's go back now that we know the 2 different options and look at which they could be. So obviously now we know why it can't be AA, because they're all AB based on how segregation happens. So we know, again, these are wrong. So it has to be AB, and it can't be C, these, because it can either be little a, little b, and big A, big B, or it can be big A, little b, little a, big B, but not the cross-contamination of these 2. So that takes out C, because that assumes that this is and not or. So the answer has to be B, and we see that, right? We actually drew this out and proved it to ourselves that this is big A, little b, big A, little b, little a, big B, little a, big B. So B is the correct answer. So this was actually a quiz question from my year, and almost everyone got it wrong because they didn't actually either A, understand the difference between meiosis and mitosis enough to eliminate the incorrect answers, or they didn't actually sit down and prove it to themselves that this was the answer. So I remember when I did this the first time, I actually had to go through and draw it out, and that was the only reason why I got this question correct. Alright, so the last topic here is nondisjunction. So nondisjunction is a little bit confusing, and because this is the quick and dirty, I'm going to give you the easy heuristic to always get a nondisjunction question correct, and then I'll go through and actually explain what the hell is happening. So, nondisjunction is the failure of homologous chromosomes or sister chromatids to separate properly during cell division. So instead of, we have our nice little segregation, yeah, like this, where like everybody's nice and happy and they're going their own merry way. Nah, we have some kind of monstrosity where like everybody, instead of going 2 different ways, everybody's just like, you know what, I'm going to the right. Like everybody goes to the right, and then that results in a cell. One cell where we have like 4, and then one cell where we have nothing. And so this is unhappy because there's too many people in the kitchen, and this is unhappy because it's really lonely. So that's like the basis of nondisjunction. We'll talk about this in slightly more scientific terms in the next slide, but here's the quick and dirty. If the result is 2 X's, then the nondisjunction happened in the mother, and it could be a nondisjunction in either meiosis 1 or meiosis 2. If you have 2 X's or 2 Y's, and there's a problem with the father, then it has to happen in mitosis 2, if there's an XY, then it has to happen in the father, and it happens in meiosis 1. So if you need to memorize something quickly before the exam and you don't want to waste time by drawing out all these things, just memorize this table and you'll be fine. But if you're like me, I want to know like why is it happening? What does this mean? So I'm actually going to go through and draw out what all these non-disjunctions mean. So obviously if you have a mother, mothers have 2 X's, then you're just drawing out the same thing with X's, so there's no point because it's going to be the same either way. So the real question, the difficult questions come when there's a non-disjunction in the father. So I'm going to focus on that most importantly. So first, actually, let's go through and look at what a normal XY segregation looks like. Okay, normal, something that I am not, because I am making genetics videos right now. Normal, this is what a normal male, oh, if you don't know what this means male, then you either need to learn more about astronomy or you need to learn it quickly because it'll be an integral part of this class. A normal male segregation looks like. So we know that males have 1 X and 1 Y because females are just pretty cool like that, and we have bar bodies. So when we have S phase, what happens? Well, now males, this is 2N, go to XX, Y, Y. Okay, so in metaphase 1, how do we know these bad boys line up? Here's the metaphase plate. If you said like this, you would be correct. Metaphase 1, then we have cells that look like this. We have XX, YY. Then in metaphase, let's just say this is metaphase 2. We're just going to skip straight there. These are going to segregate like this. And you are left with X, Y, Y. So, here's a question. Why does every single person have a 50-50 chance of being male or female? Well, when you have now children, the stork doesn't come when you make babies. You actually have 2 gametes that come together. So you know you get 1 from the mother and 1 from the father. Okay, so here's the mother. Maybe this is more basic than you need, but I always like to connect this to the bigger scale of things. Here's the father. This is not drawn to actual size. Well, we know that we're going to get an X from the mother, but here you have a 50-50 chance of getting an X or a Y from the father. So that means that this is really the only proportion that matters because you have a 100% chance of getting an X from the mother. So that's why you have a 50-50 chance of being a boy or a girl. Cool. So let's talk about non-disjunction in double Ys. So that's going to require me to erase a lot of things. Erase all link on slide. Oh, that's pretty cool. I didn't know I could do that. So let's go through what happens if there's a non-disjunction that results in YY that happens in mitosis 2. Okay, so like we just talked about in the normal cell, we're going to start out with diploid XY, 2N, and then we are going to have replication just as normal. Okay, so X, Y, X, Y, 4N. But now we are looking at disjunction in meiosis 2. So if we're having a non-disjunction in meiosis 2, is that going to change what happens at the metaphase plate of meiosis 1? No, that's going to be completely normal. So this is going to look exactly like what we would expect it to look like. X, Y, Y. Segregation results in 2 cells that look like this. Oops, oh no! It took away all my annotations, whatever. You get to see this twice. Watch. Let's see how fast I can do this. I'm going to be like a ninja, like so fast. Okay, S, replication, X, Y, Y, 4 N, lining up, metaphase 1. Okay, here's the metaphase plate, X, X, Y, Y, segregating. See, that was really fast. Then we get X, X, Y, Y. This is metaphase 2. So here's our metaphase plate. So, like we said, metaphase, or 2, meiosis 2, this is where we have our non-disjunction. So normally, we would expect them to segregate like this, right? So each gamete, so this is normal. You would expect each gamete to have an X. And then here, we would expect each one to have a Y. But instead of each Y going on its merry way, both Y's get a little greedy and go to the same side. So this results in a cell that is Y, Y, and then this one has nothing. That's a sad non-cell. Okay, so this results in 4 gametes, one of which has 2 Y's. So this is normal, this is N, this is normal, this is N, this is not normal, this is 2N, and this is 0 N's. No, that's not good. So that's what it looks like when you have a non-disjunction in YY. So now you can see why that only can happen in meiosis 2, right? Because if it happened in meiosis 1, then you'd have a problem because you'd result in XY and not what you would normally expect. Okay, so let's talk about that now. Non-disjunction that results in one cell being one gamete being 2N with XY, and that can only happen in meiosis 1. So we're going to go through this again. So we're starting out with our normal 2N cell, 2N, and we have an X and a Y. Then we have S phase, so we have replication. I know, are you sick of me talking about S phase? Good, because then you won't forget it. Okay, and then we are going to metaphase 1, and these are lining up just like how we would expect. X, X, Y, Y. But instead of these going on their merry way this way and this way, all of them go that way. So we get one daughter cell that is exactly the same, right? And one that has nothing. Oh, that's really sad. So in this case now, if this is metaphase 2, then these can segregate like this, and we're left with daughter cells that look like this. X, Y, X, Y. So again, we're dealing with 2N, 2N, but obviously these are also going to have nothing because magic's not real. Sorry, kids. 0N, 0N. So yeah, that's a mess. That results in XY, XY here. That's why it has to happen in meiosis 1. That looks like I crossed it out, but I was just trying to underline it and did a poor job. Okay, so now we understand what could happen and how this works. So hopefully this video was helpful for you. These were kind of the most important mitosis topics. Also, I thought this was a pretty cool meme. So hopefully you enjoyed this video. Hopefully it was helpful for you, and hopefully you will rock genetics. Thanks. Bye.